muscle pathology in our specific mouse model of VCP disease. Next slide, please. Oh. So this slide is very familiar for you guys already. VCP, multi-system proteinopathy that affects the bone, the muscle, and the brain. And we particularly see pathology in the cytoplasm of the cells in the central nervous system, as well as the myofibers. This includes protein aggregates and inclusion bodies. Next one, please. And people have put in efforts to characterizing the VCP mutations that leads to, and then they have shown that in biochemical analyses that it leads to ATPase activity. Here on the left, you're looking at the study by Paul Freeman's lab where they purify the VCP mutants from E. coli and analyze ATPase activity, and mostly we see increase of activity. And here on the right, you're looking at the study by Raymond Deshae's lab where they are looking at VCP's activity as an unfolded to unfold GFP as a substrate. Gradually, we're losing the fluorescent signal of GFP because GFP is gradually unfolded, and it seems that VCP mutations leads to an increased activity as well. Next. As you have heard from Dr. Schufen's lab, Dr. Schufen, where generations of VCP inhibitors has been explored, and currently we're investigating in this study the novel version of VCP inhibitor CB5083 in our specific model of VCP disease. Next. Previously in the biochemical studies by Ray Deshae's lab, where they incubate together the CB with the mutant version of A232E complex, VCP complex, and they're able to normalize the unfolded activity back to the wild type level. And that becomes one of our foundation to inhibit our hyperactivity VCP complex in our rodent model back to the wild type level and see whether we can ameliorate disease pathology. Next. Physiologically, people have suggested that VCP inhibitors could be potential efficacious in the fly model. And this is a study by UCLA, Dr. Mingguo's lab, where they created fly model containing the most common mutation of VCP, VCP-R15H, and the most severe mutation of VCP, VCP-A232E, and then they're trying to characterize the muscle structure of these fly models. As you can see, they lost the green signal, which represents the fragmented mitochondria, and then they increase the cellular apoptosis and representing here as increased red signal. However, with VCP inhibitors, those are the older generations of VCP inhibitors, we see the rescue of the mitochondria morphology and reduction of the and depletion of the cellular apoptosis, suggesting that VCP inhibitors could be potentially efficacious in the fly model in terms of rescuing mitochondria morphology. Next. So here in our study, we decided to test the novel version of VCP inhibitor, CB5083, in our model of VCP disease, VCP-R15H homozygous animal, to see if we're able to see any amelioration of disease and muscle pathology. Next. First, we decided to test the CB drug in our patient-derived myoblasts. Mainly, we wanted to first see whether these myoblasts are indeed myoblasts. So we tested using immunohistochemistry with Desmond signal, and we saw both control as well as patient cells have strong signals suggesting that they're indeed myoblasts. However, and when we're looking at TDP43 location in these two cell lines, we find TDP43 is mainly located in the nucleus of the control myoblast. However, we start seeing TDP43 leaking out into the patient myoblast cytoplasm. When we are looking at on the right, the Western blot analyses of a cytoplasmic nuclear fractionation, we found that TDP43 indeed is increased in the patient derived myoblast cytoplasm, and it's more than the control cell, suggesting that TDP43 related pathology is observed in these cells. Next. Then we looked at whether these cells are able to tolerate CB5083 treatment. So we treated cells with CB with between dose of 50 to up to 300 nanomolar, and we see steady survival rate, suggesting that these cells are able to tolerate the dose of up to 300 nanomolar of CB treatment. And when we are also incubating the cells with CB for 4 to 5 days, and we found that mitofusin 2 is steadily increased because because it was known that VCP is negative. Mitofusin 2 is negatively regulated by VCP. And in addition, we also see upregulation of P62 and LC3 with increased dosage of CB5083, suggesting that CB5083 in our cell line does modulate autophagy pathways. Next. And then we decided to move on to our in vivo model. So this is a specific model that we think it's a rapid progressing model that mimics VCP disease. This is a homozygous animal that contains the most common mutation, R155H. We treated this mice from 2-month-old for 5 months with daily oral gavage of CB5083 at dosage of 15 mg per kilo. And now you're looking at the endpoint analyses. On the top, it's the body weight analysis. On the bottom is the liver enzyme analysis. And in both cases, we did not observe any adverse effect. Suggesting that chronic CB treatment is well tolerated in our animal model. Next. And in addition, we also looked at the biomarker expression of VCP in these VCP R155H mice. Here on the panel A, you're looking at the first focusing on the first column first. When we comparing the wild type and VCP R155H mouse Western blot, we see some marker get upregulated. For example, P62, TFEB, TDP43, as well as in panel B, phosphorylated TDP43. However, we found it surprising that with VCP inhibitor CB5083 treatment chronically, these markers are dampened. And we see similar trend in an aging VCP animal that is heterozygous for R155H, that these markers are downregulated as well. Next. And now we're looking at the muscle global anatomy and architecture of the myofibers in these animals. So we looked at wild type, and if we're looking at wild type H&E staining, we found that most of the myofibers have homogeneous size, and the cell nucleus are located in the periphery of the myofibers. However, in the VCP homozygous animal, we observe heterogeneous size of myofibers, atrophic fibers, centralized nuclei, and cells infiltrating into the interstitium of the myofibers. And these phenomena are largely alleviated with CB treatment, even though we still observe heterogeneous size of the myofibers, but centralized nucleus, for example, is reduced, suggesting that muscle architecture somehow is alleviated in our animal model as well with CB treatment. Next. And then we looked at TDP43 pathology using immunohistochemistry analysis. And here, when you're looking at wild type, TDP43 is again located inside of the nucleus of the myofiber of the muscle myofibers cells. However, in the VCP R155H animal, we see an increase, a global stronger expression of TDP43, and in addition, we also see cytoplasmic expression of TDP43 as well in these muscles. However, when we're treating with CB, we saw that the cytoplasmic expansion of TDP43 somehow is alleviated, suggesting that CB5083 treatment can have some function in terms of modulating TDP43 pathology in these VCP R155H homozygous animal. Next. And we also looked at the CNS pathology in these animals, and we largely did not see any effects. This suggests that potentially is because CB5083 have a fairly low blood-brain barrier penetrance. Next. I added another slide before, but so this is actually a clinical. So this slide is actually from inside of a clinical trial when CB5083 was proceeded for a clinical trial treating 85 patients with cancer, and the trial was unfortunately halted because patients started to report vision loss and blurred vision. And later on, they found that CB5083 is actually off-targeting PDE6, which is a major player of phototransduction pathway. So we decided to look whether, despite the acute effect of CB5083 in retina function, whether we see permanent damage of retina. So we collaborated with Dr. Potetsky's lab here at UCI and then looked at the chronic treatment of animals on retina function. So we treat the animal for 5 months, and then after the last dosage, we waited for 24 hours to let the drug to be washed out inside of the body, and then we record the retina electrophysiology with ERG, and we did not see any effects in latency as well as amplitude, suggesting that the retinal function remains intact. Next. So in conclusion, we found that we demonstrated that VCP inhibitors CB5083 are safe and well tolerated in our animal model for 5-month treatment at 15 mg per kilo daily dosage. And it is able to aggregate some of the, we call it biomarker expression in our animals back to wild type levels. And it has a modulatory effect of TDP43 expression. And we did not observe CNS pathology improvements. And we think CB5083 is safe for retinal function with our dosage. And with that, we would like to thank our funding resources and patients and their families. Thank you.
I am going to share my story about VCP protect against proteotoxicities. Next one. So as we know, VCP is ATPness, a function versus different pathway inside of the cells. Right now we identified more than 30 different mutations in the MSPs, and it can affect multi-systems, including muscle, bone, and the brain. Next one. And next. So VCP is kind of like very diverse in the tissue, even if you look into the brains. It first identified with the TDP43 inclusions, either in the cortex or the spinal cord, but next. But right now, we identify more evidence, identify VCP can cause other diseases like Parkinson's. So it has the inclusion, Lewy body inclusions with alpha-synuclein, but not TDP43. So it is totally TDP43 dependent. And more recently, next. There were a paper published last year identified 2 VCP family with AD, with Caltech, but without any inclusion like TDP43, A betas, or alpha-synucleins. Then we ask the general questions: what is the role of, what is the relationship between VCP and the proteotoxic proteins? So first, before I start my story, I would like to introduce a little bit more about proteotoxic proteins. So they are recognized as a biomarker in many neurodegenerative disease in the brain, and it always contains, those proteins always contain the unstructured domains, and it prompt to aggregates like TDP43. And we think those proteins are infectious. So they can spread from one region to another, and it can take place and new aggregates to form. So this process, we call it the seeding activities, or the seeding process. And generally speaking, those proteins are toxic to cells. Yes. And in the past few decades, there is a more and more recognitions of these topics. And next, let me discuss about the tool we use. So to study the proteotoxicities, we have a fantastic alpha-synuclein biosensor line, just a tool, high throughput tool that developed from Mike Diamond's lab. So they have alpha-synuclein CFP and YFP co-expressing the same cells. Next. And when we have alpha-synuclein PFF, the preformed fiber, what we call the seed, into it, it will generate the fresh signal within 24 hours. Next. So using this tool, we're doing unbiased whole genome sequencing we transduce with Cas9 and whole genome guide RNA knockout libraries. After selection, we add alpha-synuclein seeds. And it generates the fresh signal within 24 hours. And using full sorters, we separate out the FRET positive group and the negative groups. And we do DNA extractions and next generation sequencing to resolve the gene knockout in each groups. Next. So that is a basic idea. And using this screening, we identify 153 different genes that we think can have protective effects against the protein-sensitivity. So we identify VCP as one of them. Next. We did, sorry. We did the secondary validations of the screening. We select 9 different genes and doing the siRNA knockdown in the biosensor line, and we validate our hit VCP. Next. And then to confirm the effects of VCP, we also did primary neuron culture. So we simply add the seed into the media of the culture and wait for 5 days. And you can appreciate that over here, there were new form aggregates marked by alpha-synuclein antibodies. Next. And it was confirmed by the Fraction Nation Western blots that we have accumulates of insoluble alpha-synucleins in the PFF seed-treated groups. In comparison, next, when we have VCP knockdown, we have increased level of those phospho alpha-synuclein aggregates. Next. Also, in parallel with the genetic knockdown, we also use different VCP drug, the NMS, CB drug, and L. And we see all those 3 inhibitors show us the same increase. But it's not happens in the autophagy modifiers or the proteasome inhibitors. Also, the VCP inhibitions next. In the hypophysis neurons, and we see the same effect. Next. So right now we see that if we totally dysfunction VCP, it can aggravate the alpha-synuclein seeding. So we would like to see what the effects of VCP disease mutations. So as we know, the VCP disease patients contain different kind of inclusion, including alpha-synucleins. And from our previous paper, we show you that the disease mutation actually impact the aggregate clearance, for example, the POD-Q clearance. And also VCP mutations specifically affect the lysosome autophagy pathways, which is one of the protein homeostasis pathways that involve in the aggregate clearance. So we have some clues that, okay, maybe VCP mutation doing something over here. So next. We overexpress VCP different vector with cherry tag in our alpha-synuclein biosensor line. And you can see in the left panels, in the VCP overexpressed line, you see the increase of alpha-synuclein in those 3 disease mutations overexpression, but not the wild type. Also, we repeat the same results in the VCP R158 heterozygous hippocampus neuron, and we see tons of alpha-synuclein aggregates over here, compared with the wild type sibling controls. Next. Then we ask, okay, can we see the similar effects in vivo? So we did alpha-synuclein injection into the brain to the 4-month-old mice, and then we wait another 90 days. And in the 57 control, we can some moderate alpha-synuclein aggregates throughout the brains. So here is a cortex area, but it happens throughout the brain as well. And in comparisons, in our two different VCT disease models, we see significant increase of alpha-synuclein deposit. Next. And it's quantified throughout, in different areas in the brains. Next. Here we're showing that VCP have the effects of alpha-synuclein seedings in vivo and in vitro. So then we ask if VCP have similar effects on TDP43 seeding, since most VCP patients right now have been identified with TDP43 infections. So we have the TDP43 Fred biosensor line, which is the same idea to have alpha-synuclein 1. And when we put down the VCP NMS drugs into the sensor line together with TDP43 seeds, we see the increase of TDP43 seeding activities quantified over here at different doses. And also, when we overexpress VCP disease mutations, and we also see increase of TDP43 seeding activities. Next. And also, I generate new platforms of TDP43 seedings in neurons. You can go for my papers. I did a toss of validations to make sure that it can phenocopy some of the TDP pathologies we see in the brain. And using this model, using these platforms, I compare the TDP43 seedings in our VCP R150H heterozygous neuron versus the Y-type one. And you can see the increase in seeding as well. So it gives us some evidence that VCP is not just involved in one kind of protein seedings, but also the TDP43 one. And finally, I would like to talk a little bit about the tau seedings. We did some very preliminary data on that. So we overexpress different VCP disease mutations in the tau biosensor line, and we see the similar effects as TDP43 and alpha-synucleins. And there were another paper published in Science last year. They identified the new cases with AD, and in their mutations, you can see in their Fred-based assays, you can see the same effects as what we're seeing over here. And they're also doing the in vivo studies with tau injections on their heterozygous and homozygous mice, and they see the increase of tau aggregations in the brain. So, so combined with all those three different seeds, we kind of like get into the conclusions. VCP is kind of like protect against the protein seeding in general. And we saw that the inhibition of VCP can actually aggravate the protein seedings. So thanks for my lab mates, my PI, Dr. Well, and all my collaborators and the fund for the studies, and thanks for this conference to give me opportunity to share my data.
All right, thank you, Jiang. Okay, with Feng on.
Today I'm going to share some results we recently got about the P97 inhibitor can rescue some neurodegeneration features. Heard a lot of from yesterday and this morning. So for our group, our aims is to uncover the cellular effects of P97 mutation on motor neurons. And the second one is we are going to searching for potential treatment for P97 mutation-linked motor neuron degeneration. And here is some other result. To achieve our goals, we have to have a cell model to do the research. The first thing, we use iPS-derived cell to study the disease, and we made iPS cell from patient-derived fibroblast. To avoid the genomic background effect, we also correct the mutation into wild type using CRISPR-Cas9 technology. And after CRISPR editing, we get the isogenic wild type, and we compare the pluripotency of the iPS cell and the CRISPR edited iPS cell. Their OCT4 level show no difference, means the CRISPR will not affect the pluripotency of the iPS cell. And then we use the 18 days protocol to generate motor neurons. Use this method, we can generate a lot of immature motor neurons, and those neurons can be frozen and can recover at any time, and we can perform our different experiment. And after recover the immature motor neurons, we use this method to generate the mature neurons. At day 14, post the maturation culture, we get the mature neuron. Here is the morphology of the day 14 mature neurons. We can see a lot of neurons, and they are really neuron-like. And we also use the motor neuron markers to do IF staining, and we see the positive of chat and ISL1 and 2G. And the third thing, we want to know the mature neuron is really function. So we use the Maxwell chap to detect the neuron activity, and we do see signal from both mutant neurons and also as wild type neurons. And then we want to see whether the induced motor neuron can recapitulate neuron degenerations. The first thing we did is to determine the cell survival using a live cell staining assay. We monitor the neuron at day 0, from day 0 until to day 30. At day 14, the motor neuron think is mature, and we see a slight difference between wild type and mutant neurons. With the extended maturation culture, we can see the difference in cell survival become obvious between the mutant and isogenic wild type. And also we use Western blot to detect some biomarkers we are known related to P97 function or neuron degeneration. We can see the ubiquinous protein K48 shows no difference between mutant and wild type, and also the ER stress, we didn't see difference. And we see a slightly difference in LC3 level. And p-tau level at day 20. At day 20, we see the huge difference between the cell in the cell survival between mutant and wild type, and we also found the difference in phospho-tau level and also p53 level. And here, this result indicates that our neurons can recapitulate the neurodegeneration caused by P97 mutation. Next, we use proteomics to profiling the motor neurons and to see what's the effect of P97 mutation on the neurons. So from proteomics, we identify a lot of upregulated and downregulated proteins. And to validate our proteomic finding, we choose a few of the upregulated protein and use Western blot to validate. And our Western blot result is consistent with our proteomic findings. To figure out what's the dysregulated protein might participate in the cellular functions, we do the functional enrichment analysis, and we found those cellular pathways were potentially affected by the P97 mutations. Of those pathways, the main character the character of them is they are related to cell cycle, and we list the cell cycle relative proteins, and we found most are upregulated. And then we also use RNA-seq to profiling the transcriptomic on the motor neurons. Similar, we got a lot of upregulated protein, and the pathway also revealed the cell cycle related pathway was dysregulated in the disease the mutation motor neurons. And we also compare the dysregulated cell cycle protein in protein level and the RNA level. We found most of them are both upregulated in the disease mutant, the mutant neurons. And then we want to screen for the potential treatment. The first thing we tried is use inhibitor to treat the disease neurons. And we tried different concentration. And here is a good concentration after we repeat. CB503 is around 300 nanomolar. And after 6 day treatment, we can see it can rescue the neuron loss in the mutation neurons. And also the Western blot showed it can rescue the biomarker the mark change. It means the P97 inhibitor might be rescue the neural degeneration caused by P97 mutation. And next, we also use proteomic to profiling the treatment effect, because the West block we only can see few markers, but we don't know the overall effect. Use the proteomic approach, we can know the overall effect of the inhibitor treatment. From the PC, we can see after the mutant and the wild type and the mutant are separated from each other, but after treatment, we can see the sample close to wild type, and also the dysregulated protein. The upregulated protein in the mutant after treatment, it can be reversed by both inhibitors, and the downregulated also can be rescued by the inhibitor treatment. And here we list the most significant rescued protein by both inhibitors. And after treatment, we can see the protein level almost similar to the wild type levels. And here are the pathways which might be rescued by the inhibitor or not by the inhibitors. And in the future, we are going to validate our findings in motor neurons derived from more patients, and then screen for more potential treatments. And thanks for our lab members, and thanks for the QVCP group to give me the chance to show our result.
Thank you, Feng. Great job. Thank you to Cheng and Jiang as well. All right, so we ran over a little bit. Apologize for the logistical challenges, but great job, everybody. So we are in our networking session right now. We had a lot of fun last night. If you need to take a break, you can do that. But if you go over to networking on the left side there, you'll be randomly paired with other people. I did want to also note that at the top right, 2 icons over from your picture is a little arrow looking airplane thing. That's for private messages. If you have a little dot next to it, somebody may have sent you a private message with how we showed you. So I just wanted to point that out as well. Okay, this session is now closed. We will, if you don't do the networking, which I still encourage you to do that, at 1 o'clock Eastern, we will have some more abstracts, and we're really looking forward to that. There's some really good, those will be the clinical abstracts. These were just the non-clinical abstracts. So thank you again. This session is now closed.